Inside a monarch butterfly's chrysalis, a caterpillar is dissolving and rebuilding itself into something capable of flying across a continent. But in millions of these chrysalises across North America right now, something else is also happening. A parasite is using that same transformation to complete its own reproduction, embedding its spores into the wing scales of the butterfly. The butterfly that emerges will be coated in the parasite. Its wings will be smaller, its life will be shorter, and it will spend that shortened life spreading the parasite to every plant it touches. The chrysalis has become an incubator. This is the reality for millions of monarch butterflies and queen butterflies across North America every year, and most people have never heard of the organism responsible, a microscopic parasite called Ophryocystis elektroscirrha. The monarch butterfly, Danis plexippus, doesn't need much of an introduction. It is one of the most recognizable insects in North America. Every fall, millions of monarchs travel south from Canada and the northern US into the fir forests of central Mexico, where they cluster in orange curtains on the trees, waiting out the winter before making a return journey north in the spring. The monarch is an icon. It is also under siege. Less celebrated but equally deserving of attention is the queen butterfly, Danis gillipus, the monarch's cousin. The queen shares the monarch's orange wings, but is found in the southern US and through Central America and into South America. And while they do not undertake the legendarily long-distance migration of the monarchs, they are no less magnificent. They share the monarch's dependence on milkweed, its place in the food web, and tragically, its vulnerability to this parasite. Ophryocystis elektroscirrha is a protozoan parasite belonging to the group Apicomplexa. This is an ancient and extraordinarily diverse lineage of single-celled organisms that include some of the most notorious parasites on Earth: malaria, Toxoplasma gondii, and Cryptosporidium that commonly contaminates drinking water. It is ruthlessly adapted to the Danis butterflies. The parasite exists mostly as microscopic spores, and it can survive on the surface of milkweed leaves for months, waiting patiently for a caterpillar to hatch nearby and begin to feed. Ophryocystis was first identified in 1970, infecting both monarch and queen butterfly populations in Florida. At the time of its discovery, researchers noted the parasite with scientific interest but little alarm. It appeared to be a background feature of the Danis biology, present but manageable, unlikely to pose a serious threat. Decades of subsequent research have proven the initial assessment far too optimistic. Ophryocystis is spread almost entirely through the milkweed plant itself. Adult butterflies infected with Ophryocystis are covered in spores, and as they feed on milkweed nectar, mate on the milkweed leaves, and lay their eggs on milkweed stems, they deposit spores onto the plant's surface. When a monarch caterpillar hatches from its egg, it eats its own eggshell for the first nutritional boost of its life. Since that eggshell was laid on a milkweed leaf dusted with spores, the caterpillar's very first meal is an infection. Even if the eggshell itself is clean, the caterpillar immediately begins devouring the milkweed leaf it hatched onto, and the spores coating that leaf are consumed along with the plant tissue. Infection is inevitable. Once swallowed, the spores travel to the caterpillar's gut and germinate. The single-celled organisms inside each spore emerge and begin to move. They travel through the blood and infect the layer of tissue that lies just beneath the caterpillar's exoskeleton. That layer will later become the wing scales, and body surface of the adult butterfly. There, the parasite multiplies. The caterpillar feels nothing. It continues to eat, grow, and molt through its instars with no outward sign of anything wrong. It is only when the caterpillar seals itself inside the chrysalis and begins the metamorphosis that Ophryocystis makes its final move. As the caterpillar's body literally dissolves and reorganizes itself into a butterfly, the parasite completes its sexual reproduction cycle, producing the next generation of spores. These new spores become embedded in the very cells that will form the emerging butterfly's scales. When the adult butterfly finally splits its chrysalis and unfurls its wings, it is already coated in spores. The parasite has transformed along with the host and emerged with it, ready to be passed on to the next milkweed plant, the next caterpillar, the next generation. The spread of Ophryocystis through the butterfly populations is extremely efficient. A single heavily infected adult butterfly can deposit hundreds of thousands of spores on milkweed plants during its lifetime. A moderately infected butterfly can contaminate multiple plants across a wide area as it moves through the landscape. There is no conspicuous sickness to warn other butterflies away, no change in the milkweed's appearance to deter egg-laying females. A female monarch, sensing a perfectly healthy-looking milkweed plant, lays her eggs exactly where the parasite needs her to. The efficiency of transmission increases dramatically when butterfly populations are dense. During peak migration seasons, monarch populations converge along narrow flight corridors. In these corridors, the same milkweed plants are visited repeatedly by dozens of butterflies in a single day. Each infected visitor leaves spores behind. Each subsequent caterpillar to hatch on those plants faces an ever heavier contamination load. Density, which ought to be assigned to population health and abundance, becomes, in this context, a driver of disease amplification. So what does Ophryocystis actually do to the butterflies it infects? The answer depends on dose and timing of exposure. At low spore loads, the effects are subtle: slightly reduced body size, marginally impaired immune function, and a small reduction in lifespan. An infected butterfly looks, to the casual observer, entirely normal. But normal is not what it is. Its body is smaller than it should be, its energy reserves are thinner, its life will be shorter, and every plant it touches will carry its spores. At moderate to high spore loads, the effects become more visible and more devastating. The critical damage happens during pupation, when the caterpillar is most vulnerable and the parasite is most active. Ophryocystis' reproduction inside the chrysalis consumes resources that the developing butterfly needs. The results can be a butterfly that emerges from its chrysalis already visibly compromised, with a shortened or malformed abdomen, wings that are smaller than normal, or a body that trembles from its effort of emergence. Some heavily infected butterflies fail to emerge at all, dying inside the chrysalis as the parasite completes its reproduction at the host's expense. For the butterflies that do emerge from heavily infected chrysalises, the damages to their wings is particularly cruel. Butterflies with Ophryocystis damaged wings are smaller and lighter than healthy individuals, but not in the proportions that flight requires. They fly less efficiently, tire more quickly, and are less capable of sustained powered flight than migration demands. Even infected butterflies that appear physically normal suffer a hidden cognitive and physiological toll. Monarch butterflies navigate by a sophisticated combination of time-compensated sun compass orientation and magnetic sensitivity, systems that require well-functioning nervous systems and metabolic reserves to maintain. Research has found that heavily infected monarchs show impaired migratory orientation, losing the precise southwesterly heading that would normally carry them to Mexico. Instead of flying in the straight, purposeful lines that characterize healthy migratory monarchs, infected individuals drift and get lost. Monarchs infected with Ophryocystis also show reduced reproductive success. Less able to compete for mates, have fewer viable offspring. This parasite can also be passed as an STD from one adult butterfly to another. The lifespan of a heavily infected monarch is significantly shortened. Adult monarch butterflies in the migratory generation typically live 6 to 8 months, overwintering in Mexico and returning north in the spring before dying. This extraordinary longevity is what makes the migration possible, and it depends on the butterfly's body fat reserves remaining intact. Ophryocystis depletes those reserves from within. Heavily infected monarchs die weeks or months earlier than healthy individuals. They either don't make it to Mexico, or if they do, they don't make it back. The monarchs are not entirely defenseless. Both monarch and queen butterflies have evolved a defensive relationship with their milkweed host plants that provide some protection. Milkweed plants produce toxic compounds called cardenolides that are poisonous to most animals that eat them. Monarchs and queens have evolved resistance to these toxins, and they sequester them in their own bodies, becoming toxic themselves. Cardenolides also suppress Ophryocystis growth, but the availability of medicinal milkweed cannot compensate for the collapse of the migratory purge. How Ophryocystis came to reach the prevalence it has today is largely because of conservation efforts. Native milkweed species in North America are mostly perennial. They die back to the ground each winter, removing the contaminated plant material and breaking the cycle of spore transmission. But in the last 2 decades, well-meaning gardeners and conservationists across the southern US began planting a non-native tropical milkweed imported from Mexico and Central America. Unlike native species, tropical milkweed does not die back. In the warm climates of Florida, Texas, and California, it grows year-round, providing a permanent, ever-renewing food source for butterflies. Year-round milkweed allows some monarchs and queen butterfly populations to abandon migration entirely. Rather than flying south to Mexico each autumn, these butterflies simply stayed, breeding and feeding on the same patches of tropical milkweed through the winter months. These newly non-migratory populations found themselves in a paradise of constant food and mild weather. Without the seasonal dieback that would normally cleanse the milkweed of spores, the plants became permanently contaminated. Generation after generation of caterpillars hatched onto spore-laden leaves. Infection rates in these non-migratory populations skyrocketed. In the eastern migratory population of monarch butterflies that travel to Mexico every fall, infection rates typically run below 8%. In the western population of monarchs that migrates to California, infection rates are higher, around 30%. But in the year-round, non-migratory populations that have formed around permanent tropical milkweed plantings in the south, infection rates have reached 70% or higher. The tropical milkweed planted to help these butterflies was inadvertently poisoning them. One of the most powerful tools in tracking Ophryocystis infection has been citizen science projects. The Monarch Health Project at the University of Georgia has enrolled over 100,000 volunteers across North America to collect parasite samples from wild butterflies. Together, the hundreds of thousands of samples collected over the project's lifetime have created an unprecedented picture of the parasite's distribution and prevalence across the continent, revealing the divergence between the migratory and non-migratory populations. If you would like more information on this project, or to get involved yourself, there will be a link in the description. The most immediately actionable response is to replace tropical milkweed with native milkweed species. Conservation organizations, native plant nurseries, and extension services across the southern United States have been working to promote native milkweeds. These native plants die back in the winter, breaking the Ophryocystis transmission cycle and giving local populations a seasonal reset. They are also, in most cases, better adapted to the soil and climate conditions of their region, requiring less water and maintenance than the tropical milkweeds. For homeowners who want to support monarchs and queens in their gardens, switching to native milkweed is the single most impactful action you can take.